Okay, we're going to go layer by layer in the epidermis, and we're going to talk about the differences between the cells and why we call certain layers specific names according to their characteristics and their location. So the deepest layer in the epidermis is called the stratum basale, or the basal layer, and like other basal layers, it's going to have the stem cells that will end up dividing and replacing the cells in the rest of the epidermal layer, or the epidermal layer. And so you can see that they're darker. The cells have certain characteristics, and they're more basal shaped or rounder in appearance. And here's another picture, and it kind of more clearly has the basal cells sort of stand out. And so these cells are generally going to be really spaced together closely, and they're going to be darker purple in color than oftentimes the rest of the epidermis, although I have to say the staining in skin and between skin slides really is highly variable, and so I can see that the deepness of the hemosiderin versus eosin will really make the different slides appear markedly different. Like this one isn't really stained that much with eosin, but it has a lot more hematoxylin in it, and so the whole slide looks more purple in appearance. But these cells in particular are going to be the cell line that is adjacent to the underlying connective tissue. So you should be able to see that border, and it's really just a 1 or 2 cells thick when you're looking at it according to the position of the cut. And the cells do have melanin in them, so they're going to receive melanin from the surrounding keratinocytes. So these cells are keratinocytes, generally speaking, but they're the stem cells that will eventually mature and grow more superficially and migrate towards the surface of the skin. Now, some important characteristics of these cells is that they have both desmosomes that link them to each other strongly, but they also have hemidesmosomes that link the layer here, the stratum basale, to the underlying connective tissue. And so those hemidesmosomes are very important for the integrity of the skin, which we'll see when we do the flipped classroom, how dysfunction in the development of those hemidesmosomes will cause a lot of issues. The next layer, if you're moving more superficially, so the stratum basale being here, and then as the cells move more superficially, they actually are producing more keratin, and they're not quite arranging that keratin yet, however, it's starting to fill up the cytoplasm. So you see a lot more cytoplasm in this next layer called the stratum spinosum. But the stratum spinosum's hallmark characteristic is these little prickly spines that stick out all around the exterior of the cell. And actually what those spines are desmosomes that are holding the keratinocytes in the stratum spinosum to each other. And those desmosomes exist in the stratum basale, but they are a lot more noticeable in the stratum spinosum after preparation of the tissue, because the preparation causes these cells to shrink, and the desmosomes are still linked to each other, and so it gives the outside of the cells a spiny appearance. And so it's more like a preparation artifact than it is in real life. The cells don't look spiny like this, but it helps us look at the cells and separate them from the other layers. I also want to note that in the deeper layers of the stratum spinosum, those cells are able to undergo mitosis, and so it's not exclusive to the stratum basale. However, as they migrate and develop further into the more superficial layers, those cells do not undergo any mitotic division. Here you can see other pictures of the keratinocytes in the stratum spinosum. This is a nice clear image where you can see the spines around the keratinocytes here. That's an artifact. And in this image here, this is a closer up view. Here's the nucleus of a keratinocyte and keratin in the cytoplasm here. And you can see these protrusions on the outer surface of the cell are actually the locations of the desmosomes. And when you look really close up, you can see the desmosomes closer here, these dense plaques, and the tonofibrils that are made up of keratin, that intermediate filament that attaches to the inner surface of the desmosome to that plaque and attaches that cell to its neighboring keratinocyte. So it's pretty cool characteristic that you can see histologically. The next layer of the epidermis, moving more superficial, is the stratum granulosum. In this more low-power view, the stratum granulosum is really the contact point between the stratum spinosum and the next layer, the stratum corneum, that really doesn't look very cellular at all. And so it's the last layer of the cells where you see nuclei when you get closer up. But there are some characteristics of the stratum granulosum that really makes it stand out from the other layers. And most markedly, the fact that it's usually mainly darker in color to a purple spotted appearance, depending on the clarity of the histology slide that you're looking at. And these darker granules inside are actually called keratohyalin granules, and they release, at a certain point, they will release their contents into the cytoplasm, and the filaggrin that's released will actually cause organization of the keratin that cell has been producing, and it will result in cornification of the cell. The keratin will completely organize and fill the cell up. And it takes about 2 to 6 hours. And here I've sort of highlighted that region. Let's see if I do have another slide where you can see more, a very nice clear slide where you can see the transition between the stratum spinosum and the stratum corneum. And you can see it sort of like has purple spots in it, and those are the keratinohyalin granules. If I move back to this previous slide where I've highlighted that line, I want to point out that the reason why that this layer is the last layer that has nuclei and kind of looks cellular is because during this transition of cornification, the cells also undergo apoptosis, which means that they're going to have a lot of changes to the nucleus. It's going to disintegrate eventually, and it actually even extrudes the inner contents of a lot of the organelles that were inside to the outside of the cell, but still maintains the integrity of the cell, which is a pretty cool concept that instead of undergoing apoptosis and being removed by white blood cells in the body, these cells maintain their integrity and even their desmosomes connecting each other. And so here you can see scattered nuclei. The cells will undergo organelle extrusion, and that organelle extrusion is very important for maintaining and creating a lipid-rich extracellular environment that will become the water barrier of the skin. And so it's a really important process that occurs here. And so that's the stratum granulosum. The next layer is a stratum corneum, and even from far back you can see there's definitely something different about this layer of the skin. And if I go to the section before, you can see that this layer kind of looks amorphous. It doesn't have any nuclei located in the cells that you can visualize, and it looks markedly different from anything in the rest of this slide. And if you want to look here more closer, you can see the lack of nuclei because these cells have extruded their nucleus and organelles, and it's still maintaining integrity based off the integrity of the desmosomes. An acidic environment will break the desmosome bond, and it will cause these cells to slough off, which is why people get acidic washes on their skin to make the outer layers of the skin fall off and have sort of new rejuvenated skin grow in is a very common sort of a beauty process. And so the stratum corneum is the one that has, oops, I'm sorry, I'm going backwards here, is the one that definitely is going to be beyond the water barrier. So you can still have this part of your skin sort of bubble up and wrinkle when you go into water for long periods of time. But because of the stratum granulosum underneath in that interface where the lipid-rich environment is created, you're not going to have any of the underlying layers sort of peel off or fall off based off of soaking in water. And this layer can actually become thicker if you have increased friction. It will stimulate increased activity in this epidermal layer, and you can get calluses in the stratum corneum that will build up. Now, I wanted to point out that in thick skin, you have a special layer in the stratum corneum at the base where it interfaces with the stratum granulosum, and that's called the stratum lucidum. There aren't a whole lot of characteristics of this layer that are different. It just looks somewhat clearer and lighter in appearance. It doesn't really have any different functional appearance, but it has a different kind of keratin in there that is really temporary, so as it matures then it changes, and so it gives it this clearer, lighter appearance. Okay, that's all we have for learning the different layers of the epidermis. Now we'll have a couple questions and then we'll carry on.